Now that we know how the male gametes or sperm and the female gametes or egg are formed in flowers, we can proceed in examining the process of pollination and double fertilization in plants, which are the key events that lead to seed formation. An overview of gametogenesis in flowering plants shows us that both male and female gametogenesis involve spore formation and gamete development. These two processes are referred to as microsporogenesis and microgametogenesis in the case of male gametogenesis, and megasporogenesis and megagametogenesis in the case of female gametogenesis. Again, this is in reference to the relative sizes of the male and female gametes produced by these processes. The product of male gametogenesis is a pollen grain composed of the vegetative cell and the generative cell. The division of the generative cell will give rise to two sperm cells, which are the male gametes. On the other hand, female gametogenesis produces an embryo sac contained within an ovule in an ovary. The embryo sac contains 7 cells, namely 1 egg cell, 2 synergids, 3 antipodals, and 1 central cell. The egg cell will be the cell that fuses with 1 sperm cell to produce the zygote. The synergids are the cells that flank the egg cell. They provide several purposes during fertilization, one of which is to produce attractants to guide the pollen tube to the egg cell. Now, across the polar end where the egg cell and the synergids are located, you can find the antipodals. These cells also provide several functions during fertilization. Lastly, we have the central cell. This is the only cell in the embryo sac which is diploid. It is also the only other cell that will fuse with the other sperm cell to produce the endosperm. For fertilization to occur, the mature pollen from the anther, which is the male reproductive part of the flower, needs to reach the ovule in the carpel or the female reproductive part of the flower. The physical transfer of pollens that are released from the anther to the stigma of a carpel is called pollination. Flowers can either be self-pollinated or cross-pollinated. Self-pollinated flowers have structures or mechanisms that ensure the transfer of pollen to the carpel of the same plant. Cross-pollinated flowers, on the other hand, rely on animals or the wind for the effective transfer of pollen from the anther of one flower to the stigma of another flower. During pollination, the stigma of a flower provides pollen grains a landing zone and entrance into the ovule. To be efficient in this role, the stigma has evolved structures and chemical components that are adapted to receive and trap pollen. The illustrations in the right show you examples of these structures across different plant species. These are the structures that look like hairs, just like what you have here, flaps like this one, and also tentacles. Again, these structures serve one important purpose, and that is to capture and trap the pollen. Like the stigma, pollen grains have also evolved to survive the transfer from the anther to the stigma. Remember that we are dealing with very tiny particles here that contain cells that are required for the propagation of the species. From our previous lecture, we know that the pollen has both an internal and external wall known as the intine and exine respectively. The intine is the inner wall that completely encloses the pollen grain. It is made up of cellulose and arabinons, but the main component is callose. The exine, on the other hand, is the outer wall which contains ridges, spines, and apertures which serve as a site for pollen tube germination. Deposited on the surface of the exine is a layer of extracellular matrix from the tapetum. This layer is called the pollen coat and is composed of waxes, lipids, proteins, and aromatic compounds. The pollen coat has several functions, one of which is to facilitate the dispersal of pollen from the anther. It also protects the pollen from dehydration and UV radiation as soon as it is released from the anther. The composition of the pollen coat also allows it to stick to the stigma upon landing. Lastly, the composition of the pollen coat interacts with the extracellular matrix lining the style and contributes to the tube growth. So say, for example, we have successful pollination of a flower, meaning a pollen grain has successfully landed on the stigma. What is the first thing that needs to happen for fertilization to occur? First, The pollen grains need to rehydrate or absorb water. Recall in our lesson on male gametogenesis that pollen grains undergo a highly controlled process of dehydration during maturation. Dehydration partially arrests various metabolic processes in the pollen until it is ready to germinate. In addition, the controlled desiccation of pollens, in addition to the deposition of the pollen coat, serve to protect the pollen grain from further dehydration while in transit from the anther to the stigma. As I said, excessive drying can dry up and kill the sperm cells. So when is a pollen supposed to germinate? After pollination or landing of the pollen on the stigma. So as soon as the desiccated pollen comes in contact with the stigma, compatible pollen grains adhere to the stigma, hydrate, and then grow a pollen tube. Before contact, pollen grains have an elongated shape and are relatively desiccated, as you can see in these illustrations in Figure A and then also in Figure B. When the pollen is captured by the stigma, the outermost layer of the pollen grain is deposited on the cells on the surface of the stigma, this structure right here, which is known as the papillary cells. After this, a structure known as the adhesion foot forms at the cell-to-cell interface between the pollen grain and the papillary cells. With the pollen securely attached to the stigma, it can then start rehydrating. The rehydration of pollen grain is facilitated by the release of water from the surface of the stigma. This occurs the instant the pollen coat touches the surface of the stigma. The mechanism for water release from the stigma has not been fully determined, but the action of specific genes like the aquaporin-like genes and flavonols have been implicated in the process. Pictured on the left is a scanning electron micrograph showing desiccated pollen grains landing in the stigmatic surface of the Arabidopsis plant. So if you look at these elongated structures here, this, and this, these are pollen grains. Now, do you think pollen can germinate in the surface of any plant tissue? For example, under natural conditions, pollen from a flowering plant can be blown by the wind to land on the surface of leaves. If the leaves are moist, which is the perfect condition for the pollen to rehydrate, do you think it will be able to germinate? The answer to that is no. Pollen grains do not normally respond to contact with non-stigmatic tissue, such as the leaf epidermis of an Arabidopsis plant, as shown here. The globular structures that you see here are pollen grains just sitting on the surface of the leaves, and they are not germinating. A major component of plant tissues is long-chain fatty acids. These long-chain fatty acids are responsible for restricting water flow across tissue boundaries. In this case, even if the pollen grains are sitting on the surface of a leaf, the long-chain fatty acids prevent the release of water from the leaves to the pollen. This is true for all plant organs except for the stigma. The stigmatic surface may be unique in its capacity to interactively change its water permeability on contact with pollen through alterations in long-chain fatty acid synthesis. Now on the right is a picture of pollen grains on the leaves of a mutant Arabidopsis. This mutant is deficient or is not able to produce an enzyme that synthesizes long-chain fatty acids. Because it cannot synthesize or produce long-chain fatty acids, water flow is not restricted. In this case, pollen in the surface of the mutant leaves are able to germinate as indicated by the white arrows. But again, this is a mutant. Under natural conditions, pollen grains are only able to germinate in the surface of the stigma. After rehydration, the pollen grain germinates. The vegetative cell or the tube cell leads the formation of the pollen tube. This structure that you see here is the pollen tube. In most flowering species, the generative cell divides into two sperm cells after the pollen tube starts elongating. The pollen tube elongates in the style to reach the embryo sac. As the pollen tube elongates, the cytoplasm of the vegetative cell is concentrated at the tip of the pollen tube, and the part near the grain is blocked off by callose plugs, as indicated by these white structures that you see right here. What is the purpose of these plugs? The pollen tube is the vehicle that transports the sperm cell to the embryo sac to unite with the egg cell. That means the pollen tube needs to elongate and travel down the style to reach the embryo sac. The movement of the cytoplasmic contents at the tip of the pollen tube concentrates the cellular activity to the end of the pollen tube so it can elongate. Vesicles containing cell wall materials are transported via the exoskeleton to the growing tube tip, and cell materials are deposited to the cell wall by exocytosis. In the right panel is a fluorescent micrograph showing the callose plugs in elongating pollen tubes. What guides the pollen tube towards the embryo sac? In this graphical cross-section of the flower, you can see that the pollen containing the sperm cell is trapped in the stigma. It's a structure right here. Now for the pollen to efficiently deliver the sperm to the embryo sac, which is right here, this structure is here, the pollen tube has to navigate its way efficiently to the ovule. This is the function of both the style and the embryo sac. The style and the embryo sac provide chemical signals to the pollen tube that guides it towards the embryo sac. Initially, it is the job of the style to guide the elongation of the pollen tube. As the pollen tube reaches the end of the style, the embryo sac takes over to guide the pollen tube through the micropyle. Recall that the micropyle is a small opening in the integument that serves as the entry point for the pollen tube to deliver the sperm. In this figure here, where is our micropyle? It is right here. Now the embryo sac produces a number of cellular metabolites that serve to properly guide pollen tube elongation through the micropyle and into the embryo sac. I have listed a few examples here. One is the gamma-aminobutyric acid or GABA, which is a chemical attractant secreted by the embryo sac. We also have the transcription factors MIB98 and central cell guidance or CCG. MIB98 is secreted by the synergids, these two structures that are flanking the egg, while CCG is secreted by the central cell. Both transcription factors ensure the correct guidance of the pollen tube through the micropyle. The hapless gene in the pollen tube itself recognizes the chemical signals provided by the embryo sac. When the pollen tube reaches the embryo sac, the pollen tube goes through one of the synergids. The synergids then signal the growth arrest of the pollen tube. The pollen tube in turn bursts and releases the two sperm cells. When the sperms are released or shortly after it, the synergid disintegrates. What happens then to the sperm cells? One of them fuses with the egg cell to form a diploid zygote. The other sperm, this spot right here, fuses with the central cell nuclei, forming a triploid primary endosperm. The endosperm will serve as the food source for the developing embryo in the seed during germination. The two fertilizations that occur in the embryo sac, namely the fusion of one sperm with the egg and the other sperm with the central cell, is the reason why this process is referred to as double fertilization. In general, an ovule attracts only one pollen tube. Evolutionarily, this makes sense because it will be a big waste of energy and resources if a pollen tube goes to an ovule that has already been pollinated. In summary, for fertilization to occur, the mature pollen should reach the embryo sac to deliver the sperm that will fuse with the egg cell and the central cell nuclei. The stigma serves as a landing zone and entrance for the pollen to reach the female gamete. The physical transfer of pollen to the stigma is called pollination. When the pollen lands onto the stigma, the crosstalk between the pollen and stigma allows for the rehydration of the pollen, followed by its germination. Elongation of the pollen tube towards the embryo sac is guided by chemical attractants secreted by the style and the embryo sac. When the pollen tube reaches the embryo sac, two sperm cells are released. One sperm fuses with the egg to produce a diploid zygote, and the other sperm fuses with the central cell to form a triploid primary endosperm. This fusion of each of the sperm with the egg and the central cell is called double fertilization.